process of fertilization in angiosperms is unique. It is described as double fertilization and triple fusion. Double fertilization and triple fusion. It is because there are two male gametes involved that reach the embryo sac along with the pollen tube. Out of these two male gametes, one fuses with egg cell and this forms the zygote, and the other fuses with secondary nucleus. Secondary nucleus to form an endosperm nucleus. Endosperm nucleus. Since this secondary nucleus is already diploid, the endosperm nucleus becomes triploid. So two male gametes are fusing with two separate cells, therefore it is known as double fertilization. Fusion is taking place at three stages. Number one, fusion of two primary nuclei to form a secondary nucleus. In the embryo sac, there were two primary nuclei in the middle. Just before fertilization, they fuse and form a secondary nucleus which will be diploid. Fusion number 2 is first male gamete with egg cell to form the zygote, and fusion number 3 is second male gamete with this secondary nucleus. So fusion number 1, two primary nuclei fused to form secondary nucleus. Fusion number 2, first male gamete fusing with the egg cell. Fusion number 3, second male gamete fusing with the secondary nucleus. Therefore, it is described as double fertilization and triple fusion. This entire process was first described by a biologist by name Strasburger in 1884, and triple fusion in particular was first discovered and described by Navaschin in 1887. Following fertilization, a few significant changes take place in the flower. These changes can be described as post-fertilization changes. Post-fertilization changes. Remember, there were sepals and petals in the flower. After fertilization, sepals and flutter petals fall off. Wall of the ovary becomes wall of the fruit. Zygote in the embryo sac develops into the embryo. Embryo sac transforms into the seed. The integuments become the seed coat. There is an outer seed coat called testa, inner seed coat called tegmen. Nucellus gets modified into a nutritive tissue called perisperm, and the primary endosperm nucleus which is triploid gives rise to the nutritive tissue in the seed called endosperm. So these changes now take place inside the flower following fertilization. So covering the seed there is a seed coat, nutritive tissue called perisperm that is formed from nucellus, and a very important nutritive tissue called endosperm that develops from the primary endosperm nucleus. Primary endosperm nucleus gives rise to endosperm much before the zygote develops into the embryo. There are three types in the formation of this endosperm. One is called nuclear. In the nuclear type of endosperm, there is only karyokinesis. The primary endosperm nucleus divides repeatedly and gradually gives rise to the endosperm. There is no cytokinesis. The second type is called cellular. Here karyokinesis is accompanied by cytokinesis. So formation of large number of cells representing endosperm. Nuclear type of endosperm is very common compared to cellular type. The third one is called helobial. It is observed in some orchids. It is intermediate between nuclear and cellular. Initially there is only karyokinesis, and gradually karyokinesis is accompanied by cytokinesis.